This procedure begins with culturing the bait strain in media containing normal amino acids and the SILAC control strain in heavy isotopic lysine and arginine. Equal amounts of both strains are mixed and lysed by grinding in liquid nitrogen. Crude lysate is first pelleted by centrifuge and then cleared by binding to cephalose beads. Cell lysate is then bound to IgG beads. The proteins bound to IgG beads are eluted by protease cleavage and precipitated. The sample is now ready for analysis by mass spectrometry.
Hi, I'm Chao from the Loewen lab in the Department of Cell and Developmental Biology at the Life Sciences Institute of the University of British Columbia. Today we will show you a procedure for quantitative proteomic analysis called SILAC combined with affinity purification. We use this procedure in our laboratory to study protein complexes. So let's get started.
In this procedure, the bait strain contains normal amino acids while the SILAC control strain contains heavy isotopic lysine and arginine. To get started, first grow 1 liter each of the bait strain and the control strain separately to the values shown on your screen. Next, pour the culture into 500 milliliter Nalgene centrifuge tubes. Balance the load in pellet cells at 2,580 RCF. Decant the media and resuspend the pellet with 50 milliliters of cold DH2O while transferring the